My name is Rick. I'm a third-year graduate student in the neuroscience department, and I'm going to be talking today about how do you make memories stick. But first, we're going to take a trip down memory lane for me, because I want to talk about how I got into science, because it's not always clear how people get into this pathway. And I learned about science in the way that you guys are today, through textbooks in school, and I didn't really like it. I didn't think it was very fun to memorize that the mitochondria is the powerhouse of the cell. It's kind of boring, right? And so I also didn't know that you could be a scientist, that like doing science as a profession is a thing. But when I went to the University of South Florida for my undergraduate career, I learned that you could conduct undergraduate research. So I got a chance to do this science myself. And I found that really interesting, and so I took advantage of that opportunity, and I went for it and worked for neurostructural analytics. So this is a lab where we collaborate with other scientists to help them analyze data that they get from their brains from experiments. So not their brains, but the experiments that they did, right? And so we actually trace out neurons, and we use that in order to analyze their complexity. And this is the first neuron I drew. It's beautiful, isn't it? Okay, so I guess we're using the eye of the whole room, that's it. So I went from drawing neurons to actually growing neurons at Draper Laboratory. So I tried to grow neurons outside of the body and have something that's similar to a brain so we can use that as a way to test drugs and understand how the brain works. And this is a picture of the team and me trying to look really cool in a microscope on the top left after we published our first paper. So this was really interesting. It was a lot of fun. And, but now I'm here. And so what I study now is what makes a memory. Okay, so do we have any ideas of what makes a memory? Yes?
What makes a memory is basically if you put it in a big bottle of water, like a big water, and basically that moves a bit of a current in my head into my neurons, and when I look back on that memory, it's going to the same electric current.
Okay, so he's talking about that his sensory experience somehow generates an electric current in his brain, and whenever he thinks about it, it's reactivating the same thing. So that's exactly right. But I'm going to take a little bit more zoomed in, or I guess zoomed out view, and talk about something called episodic memory. So episode, sort of like the favorite episode of a TV show that you like. But instead of that, we'll be talking about, for instance, an episode of your life. So if you wanted to explain a part of your life to somebody, you would talk about a couple of things, right? So you would talk about the where. Where did this memory happen? The when, right, so the time, when did this happen? And also the what. Was it something good? Was it something bad, right? So what's really awesome about memory is that the brain uses different parts of itself in order to hold the information for the where, the when, and the what, and it brings it all together. Not necessarily one location in your brain, but it helps you experience this as one memory, basically. So lots of people are doing awesome work to study both the when and the what, or the when and the where, but I'm going to be talking about today specifically the what. All right, and I'm going to have to give you a crash course on a couple of things so I can talk about my research. So the first is acetylcholine. So this is how chemists draw acetylcholine, but I probably couldn't draw this from memory, so we're going to talk about it from this picture, okay? So when you see this, think acetylcholine. But what is acetylcholine, right? So it's a neurotransmitter, which is a brain chemical, basically, and it's released all over your brain, especially in response to emotionally salient stimuli. So that means things that have a positive or negative value to an organism that they need to pay attention to, right? And so you really have to pay attention to those things. Acetylcholine can make some neurons talk more or less to each other because your brain cells are always communicating with each other, just nonstop. Now I want to introduce you to two regions of the brain that I'm interested in, and one of them sends acetylcholine to the other one. So to orient you, this is a side view of a mouse's brain. So if you were looking at me like this, and I had a mouse brain, you would go in this way, okay? Now we're going to focus on just this purple and orange area here, okay? And then the first area I want to talk about is the Nucleus of Samus, okay? So it's a group of cells that live in the basic forebrain, and they coordinate the activity of lots of other neurons. And so what I mean by that is they can make neurons in other brain regions fire together all at the same time. And they do this in part by using acetylcholine, okay? And they're able to reach out to other parts of the brain and release acetylcholine. And one of the places that they do this is called the Basolateral Amygdala, or BLA. So this is an almond-shaped area in the brain, shown here as a triangle due to my artistic ability that's somewhat limited. And basically, it's a group of neurons that become active when something exciting happens. So this can be something good or something bad. But basically, these cells become active, and they help form the memory that's required to store this away and think about it later. Okay, so my research is to see if the levels of acetylcholine that is released from the nucleus of Samus into the basolateral amygdala affect the strength of memories. And by strength here, I mean what makes something harder to forget and easier to learn, okay? And we use this, and we look at this by using a technique called optogenetics, okay? So let me show you a video here as I crash course. All right, so it's opto for light and genetics for genetic engineering. So we can genetically engineer neurons to express this protein, or just have this protein, to where whenever we shine a laser at them, we can make them become active. So these green things here basically allow us to just control the activity of a neuron just by shining a laser at it, okay? And we can also make these neurons glow green, so that we can know which ones are actually controlling when we look at the brain later. So that's what we have here. So we control these neurons in green here, so that whenever we shine a laser at them, we can make them become active. And so the brain's doing its thing in the background, firing. And when we shine a laser, we can make all those green neurons become active. And what's really great about this is that we can see how this changes what the brain does in a wider scale by just focusing on these neurons. Okay? All right. So to recap, I am genetically engineering the nucleus of Samus neurons that send their arms to the amygdala, and I want to see if the level of acetylcholine, if that affects the strength of memories. So to show you that I can do this, here's a picture of a beautiful green neuron from the nucleus of Samus, okay? And then here is a part of the amygdala, and you can see all that green area. That's where the nucleus of Samus is sending all of its arms. And I implant something called an optic fiber, which is kind of like a flashlight, and we're able to connect it to a laser and shine light and make these neurons release more acetylcholine in the amygdala. Okay? Does that make sense so far? Sort of? Okay. So that part's really cool, but what's even better is that we can do this in awake behaving animals. So we can do all that stuff I just talked about inside of an animal and see how it changes their behavior. Okay? Now, some of my colleagues at Stony Brook University in New York, they did this for negative memories. All right? So they took control animals, which is just a fancy way to say normal mice, and they compared it to opto animals, which is where they increased the concentration of acetylcholine in the BLA. And they wanted to see how it would affect the memories for this negative experience. So they looked to see 1 day later how well the animals remembered this experience, and both groups of animals remembered about the same. But when they looked 6 days later, so much later for this long-term memory, they saw that, as they expected, the normal mice forgot the memory eventually. But when they looked at the opto mice, when they gave light only during training, whenever they learned, they saw that the memory was just as strong later as it was before. So they've changed the strength of a memory. So you can think about acetylcholine as governing the strength of the memory. The more acetylcholine there is during learning, the stronger it is and harder to forget. Okay? So they showed that acetylcholine governs the strength of negative memories. But, as I talked about earlier, the BLA is also important for positive memories. So, does that mean that acetylcholine works in the same way for positive memories that it does for negative memories? That's an open question. We really don't know. And that's what my project focuses on. And I use the same exact system to where I stimulate the release of acetylcholine in the amygdala and see how that changes in a positive memory. Okay? And we do something, we use something called an operant box. So I put this mouse in a box, and it's connected to a computer to where I can see what the mouse is doing. So I've trained the mouse by trial and error, because unfortunately I can't tell the mouse what I want it to do. I train it to, whenever this sound comes on, it's basically a beep, the mouse goes over here to this area called the nose poke, and it pokes its nose in it. And when it does that, it gets a reward, or a piece of cheese. It's not really cheese, but I put it here for now. Anyways, so, and at the same time that they get this reward, I'm also stimulating the level of acetylcholine that's released in the amygdala. So I'm hoping that maybe this will make the memory stronger. And I'll be looking at that by showing you some of the data here in a second. And so the mouse is able to get the reward, and then it can do this over and over again. So here's some of my preliminary data just for the control animals. So this is just the normal mice without the opto. So on the x-axis here, we have training day. So this is just how many times I put the animals in the box. And on the y-axis, we have number of rewards earned. So how good are these animals at doing this task? And you can see it takes a few days because, as I said, they have to learn by trial and error. And it takes about 5 to 6 days until animals get about 30 rewards per day, shown here by this green line. So 5 or 6 days, and what I expect is, if I deliver the optostim, so by increasing the acetylcholine concentration in the amygdala, I can make these animals learn faster, okay? So I'll be able to steepen this curve, which means they cross that green line quicker, so they've learned it faster. And we also do something called extinction. So this isn't dropping a meteor on the mice, like you from the last talk, right? This is just making them forget this experience. So we're not giving them a reward anymore whenever they perform the behavior. We want to see how long it takes them until they forget. And so what we can see here is it takes about 2 or 3 days until the mice stop performing, to where they've sort of forgotten. And what I expect is that if I deliver light during training, I can make these animals take a longer time to forget it, okay? So where am I at? What have I seen so far? Well, it's a work in progress. So I was just in lab today, and I'm still collecting the data to try and see if my hypotheses are correct. And that's part of how science goes. We don't have the answers right away. We've got to kind of keep working on it. But if you want to check back with me in a couple months, maybe we'll see what happens. All right, so to recap, some of my collaborators have shown that acetylcholine levels govern the strength of memories. More acetylcholine means a stronger memory. But I want to know whether or not this applies to positive memories as well. But why do we care about acetylcholine, right? Well, as I mentioned, it's released all over the brain all the time. So we can't just take acetylcholine if we want to remember something more, or take less of it if we want to not remember something, because it's released all over the brain in specific areas at given times. So for instance, if we took some more acetylcholine, it might not be so good because it's been linked with anxiety and depression. And also, too little acetylcholine has been linked with Alzheimer's disease as well. So it's a really delicate balance that we have to try and understand where acetylcholine is important in the brain and when, so we can help develop new therapies for people that have memory disorders, such as Alzheimer's or addiction. Okay? So does anybody find this interesting? Well, okay, couple, that's good. All right, so if you're interested in this, there's a lot still to be done. So for instance, one question is total recall. Right, so maybe the parents know this movie, Total Recall, or Arnold Schwarzenegger movie. But basically, some people can remember every single thing in their lives, where they knew what they had for breakfast 3 weeks ago. All right, we don't really know how that happens, but we're interested in figuring that out. And on the flip side, amnesia. Why do some people forget things? Why do we all forget things? We're not entirely sure. We're still trying to work on that. And then also, how does the brain bring together all of the components of a memory? I said that it's not in one place. But how does the brain pull all that together to make it so you're thinking about one certain thing? And lastly, how do we know that we're recalling a memory and not reliving it? So if you're thinking about what you did yesterday, how do you know that you're not there doing it and you're just thinking about it, right? You're just daydreaming or whatever. Okay? So there's some questions in the memory field that are still there that need to be answered, and if you guys are interested, it'd be really great if you join the team. So thank you all very much.
Awesome. Does anybody have any questions for Rick?
For the positive stimuli, you said you had a nose poke and cheese. What did you do for the negative?
For the negative, so I didn't do this experiment with some of my colleagues. And what they did was they had a sound just like I did, but they gave them a little foot shock. So basically they learned to associate that whenever the sound comes on, they would get a little foot shock. So the way they measure that is that they play the sound and they see how much the mice freeze. Because whenever mice are scared, they freeze. So that's how the negative experience was given and also measured. Great question.
How does photographic memory work in the brain?
So that's one of the future directions that I had. We don't really know. I certainly have no idea because I don't have photographic memory, but it's a great question. I would imagine that it has something to do with the fact that people maybe forget less, because your brain, it would be really terrible for most people if they remembered every single thing about their lives, right? It would be kind of boring. I do a lot of things that I don't want to remember. So I assume that it has something to do with some sort of pruning of some memories that aren't important, and that just doesn't happen in those people. It's a great question, and it's super interesting. I think we had a question up here, too. He's coming from.
What causes you to think about something all day, even though you don't want to?
So like a song that's stuck in your head or something? Yeah, I don't know. I mean, I guess that's why they make the big money, right? They're able to make you remember something that you don't want to from the songs. I honestly don't know why it is that we do that. Maybe it's just something that's really, really catchy. So it's really easy to remember like music lyrics, but not vocab words, for instance, right? So I think part of that has to do with just that we're good at remembering just melodies or tones or something like that. But I honestly don't know why we have to do that. It's kind of annoying, though, isn't it? Yeah.
Okay, probably one more.
I think we have one right here.
At some point in the future, do you think there would be a way to make synthetic acetylcholine? If we could implant it into the amygdala, do you think people could get photographic memory or like increased memory capacity?
Yeah, that's a good question. So it's unclear if it's for instance, so you're saying like we just sort of basically inject acetylcholine whenever we want to, right? And see if we can make people have photographic memory into the amygdala. It's unclear if it works in sort of what's called a volume transmission. Like you just put a bunch of acetylcholine in the amygdala, does that matter? Or does it matter which neurons in the amygdala are getting the acetylcholine? So it's a little unclear exactly how that works, but I think it's possible that once we learn how what's called the circuit works, like the different cells that are involved, if we can figure out exactly